Good morning. Here we are again. Oh, I'm doing my pulse oximetry. I'm at 91.88. 91's not so good. But hey, let's go and look at this new paper on mitochondria that have been kind of put down as the bad guys in COVID-19, but they're not the bad guys. Hypoxia is the bad guy. Hypoxia is like, it's like we've got soldiers and we don't feed them, don't give them water, don't let them sleep, and they're, all they've got maybe, and we expect them to fight our battles for us. No. The mitochondria are warriors for us. And so this article, I have a little bit of a, medicine is disease-based. It is disease-based and disease-oriented. And since I'm gonna tank up before I do this, modern medicine is a disease-based model. Someone gets sick and we learn from it. COVID moves in and we do all this science on it, try to understand it. And yes, the mitochondria are distressed, under duress, under distress. They do produce antioxidants and all these cascades trying to fight the virus. Now, if they're already distressed, chronic hypoxia, progressive mild to moderate hypoxia, they are going to be challenged to do their job. So that's what this article, modern medicine is disease-based. And there's so many examples of that. In other words, we're going to tell you everything that's wrong. From everything that's wrong, we're going to go in and try to fix it. Rather than, let's get you on a solid foundation, optimal health, optimal parenting, support system, mentors, standard of living, basic standard of living, fresh air, clean water, proper nutrition. These aisles and aisles of Coke and sugar in our grocery stores, maybe there are too many of those aisles. We need to spread them out a little bit, because this COVID-19 is a glycolytic, glycosis, which is how life started. So disease-based. This article, before we go into life, how it started, talks about mitochondria as though it's if we get rid of the mitochondria, we'll get rid of the problem. Wow. It's just the way it's written. It's just the approach to, yes, it's the mitochondria's fault, when in essence, it's the hypoxia is the issue. And oxygenation, hydration, exercise is the solution. So it's not just getting rid of the problem, it's understanding the solution so that there isn't a problem. So the reason we have a problem with mitochondria is because of hypoxia. And I just took my finger pulse oximeter without taking deep breaths. I'm doing a lecture on the oxygen gradient today. I'm surprised it even read through this. It's an older one, and it's not even, there it is. Okay, let's see if it gives me a better reading. I've taken a couple of breaths, but I was down to 91, and you're supposed to be around 95. And I've got a polish on, which is hard for it to read through. I'm surprised it even read through it. There you go, we'll see. So it takes 2 or 3 seconds for oxygen to transfer through the lungs. Let's see if I can get it on this finger. So, we can kind of test it from the finger. Well, the finger is a very anaerobic, low oxygen tissue. So how accurate is it? is my question. And it's not giving it to me. I just happened to get it right before we went on. So basically, I need to take the polish off. Here we go. 91 again. Now, the batteries may be low, and I know this is a possibility. And I guess my heart rate is 84. That makes sense? Okay, whatever. Okay, so that's low. That's low. It's possible I could have COVID and be asymptomatic, and that's why it's low. It's an interesting question. Interesting possibility. So, meanwhile, I'm going to do this lecture because there's an article that just came out on mitochondria, and I'm going to review it for us, with us, together. And I've made some highlights on it. It's from a French, Saleh Singh Edias, and and there are three, Oman, Sultan Qaboos University Faculté de Médecine, Port-Royal, and Paris, France, and Birmingham, Alabama, Integrated Center for Aging Research. So I may contact the Alabama group today. And je peux parler français, so I may speak with the French as well. So it goes on to how mitochondria causes the cytokine storm. Mitochondria regulate the cellular functions and provide energy to according to the body's needs. Now, I talk about that in a PROoxic way, ultra-oxic way, best case scenario, oxygenated way, because appropriate mitochondrial functioning relies on oxygen and iron uptake. It has three essential activities in the iron realm. Heme synthesis, it makes the heme that holds the iron, that gets the oxygen, that takes it back, that brings it to the mitochondria, and provides the ATP. I'll have to make a song for that, children. The iron is made by iron sulfur, a cluster biogenesis. I'll have to look up the sulfur. I didn't know there was sulfur involved in there. And storage in mitochondrial ferritin. So ferritin goes and grabs iron, holds on to it until the body has a big oxygen need, and stores it when it's using oxygen. And when it's not using oxygen, iron can be toxic and it grabs it all up. Iron is not needed in first trimester. It's one of the few arenas in biogenesis, embryogenesis, where that magnetism of iron and oxygen is also magnetic. And together they are zero magnetism. You can't detect it. We're going to talk a little bit about fMRIs today because that's how they detect function in the fMRIs, is that we're thinking about reading. Here's a person, right? And they're thinking about reading. Oh yeah, I love to read. So we're thinking, I was thinking about this, we're reading. Okay, great. It's in this back area of the left hemisphere. There's a connection to the prefrontal prefrontal cortex. This is, I'm guessing from my knowledge that this is where these are, between Wernicke's and Broca's. You're going to find this reading thing. You're going to find it here, and you're going to find it, oh yes, I'll learn something new. So you're going to get a little bit in the right hemisphere where there's creativity. And so in the brain, you have to have both sides of it. You're going to have, this is the right hemisphere, you're going to have a little bit there, and this is the left hemisphere, you'll have a little bit in, around here, I would say. And then in the medial prefrontal cortex, right in the center of the brain, back, around this area. So that's my guess. And that's what the fMRI, the functional magnetic resonance imaging, will do for us. It will show us what fires. And what happens when you fire? You burn energy. And what is the energy? Oxygen. So hemoglobin gets oxygenated, and the oxygen is taken to the mitochondria for all cell function. This article goes beautifully into that the mitochondria, oh boy, it goes into all the problems that mitochondria do first, as though it's some sort of, get rid of the mitochondria. But no. Oh man, it goes pages and pages. At the very end, it says, actually, mitochondria are our best friends, our best buddies, our soldiers. We wouldn't be here. We will be sliming along the ground as we were millions of years ago if it weren't for mitochondria. That's how important they are. So, in the fMRI, big machine, right? Big machine that we slide in and we sit there quietly, don't move. And this big, huge magnet that's looking for magnetic signals. Goes all over looking for magnetic signals. And that magnet, guess what it's surrounded by? Copper. Copper protects the integrity of the signal. That signal's not going anywhere but into the computer. If the copper weren't there, it would fly into outer space. So that's what the copper does. It protects. This is copper, Cu, and this is a magnet, very, very powerful magnet. So iron is magnetic. Iron, we know that. Iron is magnetic. It's got a negative charge, and it's a 2 negative when it goes and picks up the oxygen. Keep going back and forth on this. Oxygen has a positive charge, and they stick together, and they demagnetize each other. There's no magnetism there. So that's what the magnet cannot see. The magnet cannot see when there's iron and oxygen are stuck together. When they're stuck together, it's blind. It can't see it. But when oxygen is used in the areas of the brain we mentioned, it peels off, goes to the mitochondria, creates the function, and the iron is loose again. I think it's in a 3 model, but it's still got a negative charge, and the magnet picks up the deoxygenated hemoglobin, the hemoglobin that has lost oxygen due to function. And you'll see those 3 areas in the brain, if those are the areas, you'll see them light up. It's very exciting. It's very amazing how that works, but it picks up the oxygenation. So, it's very counterintuitive. It's kind of fun to understand, when you see something that looks obvious, like the mitochondria are the problem. Guess what? The mitochondria aren't the problem at all. They are the solution. Those are the people who are not getting it, are the ones who have healthy mitochondria. So, I think this paper should make that clear from the very beginning. Mitochondria are our friends. They are the warriors. They are the rescue. They are the fortress. And if you're hypoxic, you have disease, you are overweight, you have all those things, and the doctors are trying to make a pill or a vaccine. God help us if they go after the mitochondria and try to disable the mitochondria. Please don't do it that way. Please don't do it that way. And then they'll give you another pill. I'm so grateful I went to Hopkins. I'm so very grateful I'm a Hopkins graduate, because you know what? Bless their souls. Hopkins, there it is. I had a great year there. Hallelujah, Hopkins. They look at the solutions, no matter what the cost. If it costs them $100 billion to say mitochondria are the answer, not the problem, they'll do it. That's integrity. And everyone should have that kind of integrity. Because at the end of the day, we want to live and thrive and evolve within a human lifespan. Excellent. So let's go back to our paper. Let's see. What happened to it? All right, I'm kind of at the end of it, but it's fascinating in that it says during hypoxia, just like the Nobel 2019 prize in physiology, hypoxia is the cause, not the mitochondria. Hypoxia is the cause of mitochondria distress. And this article goes on and on and on. I'm going to put it in the notes today. It talks very much about how, talks a little bit about manganese. Reduced mitochondrial respiration, that means hypoxia, causes abnormal metal distribution, including manganese, copper, and zinc. Bingo. Bingo. Reduced mitochondrial manganese, that's why I'm taking manganese, can result in mitochondrial dysfunction because they're even more starved because manganese pries oxygen out in emergencies for them. If you don't have manganese, you're reliant on the lungs. If you've got COVID, you're out of business. This is due likely to decreased activity of mitochondria manganese-dependent superoxide dismutase. This is an enzyme that protects mitochondria from respiration-generated free radicals. So when you're hypoxic, mitochondria create free radicals, including superoxide dismutase, to manage the hypoxia, and the manganese dependent superoxide dismutase is also an enzyme that protects it from the free radicals that it produces. Holy moly. So anaerobic respiration, hypoxia, anaerobic respiration, they make it sound fancy, but it's really hypoxia, makes the pyruvate that we've talked about reduce into lactate. That is not good. We know lactate is not good. Susceptibility of loosely bound iron, and iron just goes free range. So just like we've been saying, this article confirms what we've been saying, that manganese helps us by prying oxygen out of water during periods of distress. That could be lung problems, lung issues, as we have, concussion, where there's inflammation and flooding in the brain. It's the same thing, ischemia, edema, swelling, stroke, etc. Blood clots. Ay, ay, ay. Okay, so that's the manganese. The copper, mitochondria. This is not modern medicine. This is our future medicine, please. Medicine in light of COVID-19 is mitochondria. Health. If we can work it out, we can make this happen. Mitochondria health, because mitochondria have manganese. In fact, they're created by manganese, iron, right? Manganese plus iron plus oxygen plus fluid. It generates fluid. Plus light, that's how it kind of has come into us. I'm not sure where the light is in this, but I'm sure it's in there. Zinc and copper come together to make light. So copper protects cells. You can find it just like it protects the magnet in the functional fMRI. It protects cells too. And so when mitochondria If they don't have if they don't have manganese, the mitochondria go down. Right? That's the mitochondria go down because they can't cleave oxygen. They can't do that without manganese. So, and where there's copper loose, unregulated, you're going to have blood clots. Copper is responsible for blood clotting. And now we're going over to zinc. These two are very involved in first trimester embryogenesis and stay with us till we're about 30. And then zinc is locked away for emergency, seek and destroy. And copper continues to protect the integrity of what we have, how we are growing. So copper is loose. It's loose. And zinc is less released because because mitochondria, I should put that in red. Mitochondria keep it locked up. Mitochondria hold the key to letting zinc out. And when there's less zinc, you're going to get these foot lesions and other things, and the skin rashes and things. So, and that makes sense. And guess who's getting the skin rashes? Remember I said that zinc is from Tri1 to about the age 30, depending on if you're male or female. It's about 28 for females, a little later for men. Zinc creates the rashes. Okay. So that's it's very interesting science, and this article has done a good job at doing part of it. My problem with this paper is that it makes it sound like mitochondria is the enemy. Hypoxia is the enemy. This result, these things, give solid ground to consider the role of mitochondria in COVID-19 illness, which leads to respiratory failure. No, the role of hypoxia, which leads to weak mitochondria and contributes to the insane cascade that enables COVID to run rampant in a biological system, including respiratory failure. So a dysfunctional mitochondrion would result in iron accumulation due to its incapacity to metabolize iron. How does a mitochondria metabolize iron? How does it do that? Well, here comes the iron with an oxygen, and it metabolizes it because it drops off the oxygen, and it releases an electron that iron takes up to become Fe3, and then iron goes off to get more oxygen. That's what it's talking about. Its incapacity to metabolize iron. I would say a dysfunctional mitochondria is the result of hypoxia that leads to iron accumulation due to the mitochondria's incapacity to metabolize oxygen. And so in COVID-19, these mitochondria are engorged with electrons. These electrons just build up because there's no oxygen, and the iron is coming around in this form and probably staying in this form. I haven't seen the information on that, but I would imagine the iron in the blood is unbound to oxygen. That makes, of course, it's unbound. And it's not getting the third electron. That would be my guess. It's usually iron with an extra electron. Oh gosh, this is kind of a little confusing. I'm not my expertise in this, and it's O2 plus. So when it gets the oxygen, oh God, I thought it was the other way because I know that however it works in here, but my guess is that the iron is building up is the one that is, not linked to oxygen and does not have the extra electron. And that's a different kind of beast. These one electron, more or less, really changes the function of a lot of things, especially manganese. I'm still trying to get these straight, but down. So, when the planet became oxygenated millions, billions of years ago, increased oxygen, whoopee! And at the time, there was a lot of life on the planet, all kinds of things, worms and mushrooms, or maybe a precursor to mushrooms, maybe insects. I don't know. That's a whole different science, but small animals, a lot of small animals, small algae, amoebas, little things. Viruses are relatively recent, from my understanding. What does that tell me? I should say their discovery was recent. They've probably been around for a long time, but and I'm no expert on viruses, but I am, following this COVID-19, what it does, what it doesn't do, and looking into it. So and before that, before oxygenation, we had a low oxygen environment. Yeah, low oxygen. And that's where all these wonderful little single cell doobobs were hanging out. Little worms, maybe baby mushrooms, because fungi are oxygen dependent too. You can drown a mushroom, believe it or not, and all sorts of other properties. And our brains are very much like that, mushroom cells. And amoebas, algae, and other very small, tiny little creatures, very tiny, that survived a bump in oxygenation. These guys survived it, but they survived it add mitochondria, which are metals-based bacteria. Biogenesis, organogenesis, and manganese at the bottom of the ocean. Oh gosh, at one point it creates material. I'm not sure if they're losing or gaining these electrons. I have to brush up on all of that as I keep going here. It's a lot of stuff to understand. But at the bottom, it decomposes, but I thought it also composes as well. One of these stages, you add sunlight. This one does it splits oxygen out of fluid at a lower deep. This is deep, mid sea, and this is surface, but still covered by water. And this one with sunshine, I guess this is where the material is built, builds material. That makes sense. I'll double check that. I thought it was at the bottom that it decomposes. So it degrades material when it's low oxygen, no sunlight. Sound familiar? I mean, these things all causal. So mitochondria came in with these guys and made, four-legged creatures and giraffes and, whatever, camels. And us, we're oxygenated. We're highly oxygenated. We've stood up. We have a very refined visual system. And it just all comes together that oxygenation, PROoxic practices keep the virus away and keep us evolving. In the best case scenario, the optimal, Optoxia. We'll talk about that today. I was going to get into ultroxia and establishing an oxygen gradient. There are so many mitochondria in a cell. I need to make them purple as I usually do. There's so many mitochondria in a cell, they all need oxygen. And they're all doing like supersonic. It's like having all these turbo pistons in an engine. You don't just fire one of them to get the engine going. You fire them all. So what we need and what we're going to talk about today in our PROxia series is establishing an oxygen gradient. So basically, you breathe 10 times before you even move to do something. And you're like in the fMRI, I'm going to read a really complex, wonderful book. One page at a time, yes. And I've read books before, so I'm going to use that memory system. I'm firing up my reading pathways, which is between the Broca's area and the Wernicke's area. I'm not exactly sure, but I think that's about where it is in fMRI studies. And I'm going to learn something new and novel, and I'm going to grow as a result of it. My frontal cortex is going to grow, and it's going to combine with my feelings and other things. There's a lot that goes on that the fMRI would fire. So we are fueling the initiative, fueling the directive, fueling the upcoming processes, the upcoming actions that include putting the book in your hands, enjoying the weight of the book in your hands, taking a breath before every paragraph, enjoying the full content of every paragraph, using your imagination as well as, while you're piggybacking on the author's imagination. It becomes a massively enriching and enjoyable and flow concept. Flow. So in order for that to happen, those 10 breaths establish an oxygen gradient in the area. Now, in today's fMRIs, basically what they're studying, I know very little. They say relax, listen to the music, but they don't, every study that I know of should be controlled for oxygen levels. Absolutely. Is there an oxygen gradient? Are they relaxed? All right. That's normoxia. Are they hypoxic? Are they coming in, oh my God, I don't like this thing. I'm barely breathing. So this is an oxygen gradient. There's lots of oxygen. Oxygen. And the cell, the reading cell, let's say this is a cell with all its, things to memory and things to vision. And so the signal comes along, and this cell is responsible for, reading T's. There's a group of cells that reads a T. Oh my gosh. And so that's how it goes boom, 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 in light of an oxygen gradient. Okay, so that's the oxygen gradient. Yay, it's a good thing. The oxygen gradient is our friend. Yes. We want the oxygen gradient. Now let's look at a typical a normal fMRI, and they just have the same cells, and they're in a big old rattly machine that makes a tremendous amount of noise, a lot of noise pollution. They put some earplugs on or earphones and they play some sort of rock music, whatever, and you're supposed to think while you're listening to music. They usually have music, and if they don't, it's just a lot of noise. And so there's no real, it's just kind of normal. This is ultroxic. I call this ultroxia. Ultroxia. And this is kind of normoxia. And so there's no real oxygen gradient. It's just there's oxygen all over the system, and it's floating around, da 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 da. And so there's a little bit of oxygen just passing by in a cell. We haven't really thought about doing anything. Here we've thought about it. Reading is a very specific thing, and we can increase. It's like when we oxygenate playing tennis. I'm going to play some tennis. First thing I feel is my neck and shoulder, because that is a shoulder game. If I'm thinking bike riding, it goes in, these muscles activate when I'm thinking about what I'm going to be doing. And I'm sure it will do that to you too, to the extent that you're fueling the imagination to pre-engage these muscle sets. This is PROoxia outcomes. And most of us are hypoxic. So we have to reverse that. And I'm writing a book on that. I spoke with a publisher yesterday. It's gonna be all good. It's a good time for me to put my work out in this field. So here's the cell in the normoxia. It's got all the things too, has an experience with it. Now, if you have a history of oxygen gradient, man, you've got all sorts of things going. You've got imagination, you've got creativity, you've got, I'm going to write my own story. You've got, there's an oxygen gradient cell trained with an oxygen gradient, enriched environment, and it's enriched because you're present, you're in the flow, you're wow. I so enjoy reading this book with this table here, and there's such a breeze outside. It's kind of going with me as I read the book. I'm in the flow of reading this book. Oxygen gradient. So this is kind of normoxic, which can turn to hypoxic. Oh my God, what are they seeing? Am I really reading? Oh gosh, I'm not sure if I'm reading or not. You get all kind of epileptic. Hypoxia. Okay, so here's the cell there, and this cell, the signal comes, and I said the signal came over here. We looked at this when the signal comes to the oxygen gradient cell, it fires and fires and fires and fires and fires and fires. It repeated fires. It can fire repeatedly. Yeah, great. Turbo. Over here, because there's not very much oxygen, it's just kind of floating around, it fires and then it goes down in oxygen level. It deoxygenates, and it takes like up to 15 seconds to re-oxygenate. So it has to kind of help me. It kind of has to pull it in. It has a resting kind of some function, and there's antioxidants released. There's oxidative stress. There's any number of things that start to cascade. And if you keep pushing the reading, keep pushing it on this, the signal sort of peters out. Well, we have to find other cells to do it. So this is the importance of oxygen. And if there's low oxygen, if there's hypoxia, and the cell is, it just barely, it's more, oh gosh, you get a one shot, half a shot from the cell. It backfires and moves a couple feet and then says, find another vehicle. Something like that. So hypoxia is just not good. We want the oxygen gradient. I'll be talking about that on my session today in PROoxia. And so mitochondria have enabled us to evolve. I keep stressing that, and COVID-19 is not enabling this. COVID-19 is the opposite of this. So when we don't have so oxygen gradient, fueling the directive, getting in the flow, fueling the system, evolve. This is evolution, oxygenated evolution. This is surprise, surprise. Oh, it's magnetic. There you go. This is surprise, surprise. Boom, you made us fire. Now we have to repair, so we can fire again. And it's going to take a long time. And we'll send a message to the DNA that this happened. And here, man, your DNA is ready. Your DNA is ready for change. And there's no damage. This is damage. This is kind of a damage zone when it depolarizes. And so it doesn't depolarize where there's an oxygen gradient. That's evolution. That's optimal evolution. That's our ability to evolve within a lifespan. It's very exciting, and I'm very excited to share this with you. There's so much more to talk about in this realm. PROoxia, Ultroxia, Primoxia. What else? There's so much to talk about. So COVID, all right, let's leave the note. Okay, we kind of understand. I hope you've got that. COVID works on a glucose gradient. In the presence of a glucose gradient, COVID seems to have a field day. Pyruvate turns to, so glucose creates pyruvate, turns to lactic acid. We know that's not good, right? Destroys, it's destructive. COVID moves into our cell. Mitochondria move into our cell many years ago and builds it bigger, stronger. Simone Biles, all these people, you know. This is mitochondria. Yay! Stars. Yes, we're just growing and making humans better, smarter, potentially. And then industry has destroyed our oxygen environment and we're boxed in plastic. That's not good. So COVID moves in and, gosh, what was I going to say? It feeds on our glucose. So it's just moving in on a flipped evolutionary trajectory where we have prioritized glucose above hydration and oxygen. So the gradient in COVID, I'll put it in a nice dirty brown because that's what it is, you know. Oh, I was going to say, so this is glucose. Glucose. Yep. COVID says, gimme. I like the glucose because mitochondria builds a cell, builds tissue, creates, sustains, fuels, bolsters, turbos, lets us go and do and grow and specialize, flow. athletes, cardiovascular. That's mitochondria. Mitochondria has its own DNA. DNA. Yay, it's self-contained. It has almost like has a mind of its own. The virus has an RNA, has half a DNA, RNA, ribonucleic acid, viral. Only half. It's only a half. So this moves into a cell and it starts eating it up. Yum, 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 yum. So it decomposes in order to survive. It cannot survive outside of a host. Mitochondria can survive. Look at Gallionella. Man, it's all over the place. Gallionella is some crazy metals-based bacteria. Mitochondria survives and grows and thrives just fine outside of humans in the form of Gallionella. COVID-19 eats, devours our cells. It goes in as RNA and it steals our messenger RNA. It steals it. It says, I need this, and I need this, and I'm going to grab this protein, I'm going to grab this vesicle, it just goes in and loots our cell and destroys it. And that is a hypoxic, overglycolytic, weak mitochondria. Mitochondria are disabled and weak cells and hypoxic cells. Oh my goodness. Okay, I guess I may seem to be repeating myself, but there are many stories to tell in this, and so COVID devours our cells. Mitochondria build, protect, enable, sustain, bolster, colonize, fight for our cells, put us together at night, clean us up at night. And COVID eats and destroys and fuels through glucose. Glucose is all over this. Hypoxia and glucose. So this isn't opportunistic. I know there's one other thing. All right, I'm here. I'm going to keep talking because there's one other thing I wanted to talk about. Back then when oxygenation came about, there were two forms of life that came out. One was, mitochondria, oxygen, diurnal, daytime, sunshine, intelligence, I don't put a brain in there, species, humans. And another were things like bats, and worms, and naked mole rats. They study those a lot. Really creepy things that live in hypoxic conditions. So these were things that could handle the mitochondria and survived, and the oxygen. And these were things that couldn't quite handle it. And I think they still have some mitochondria, I don't know, but they live in a low oxygen arena. They have, they're anoxic. And bats, worms, civets, naked mole rats, others. Now there's some maybe who have adapted, like turtles. All animals should be reevaluated in light of their oxygen needs, their ability to survive in low oxygen conditions, and many other aspects of biogenesis. So it's very exciting. I'm going to cut off there. We'll come back to these animals because it's no coincidence that COVID-19 emerged out of bats, civets, and it wouldn't surprise me if it would have a field day with naked mole rats. Be curious to see if COVID would, let's not even, play around with that sort of stuff, but I'm saying that COVID comes from these nocturnal, they come to life at night, echolocaic um, hypoxic level, twitch level animals. Very interesting science. Let's keep going with it. Thanks for being here. Thanks for working with me and listening. Have a good day.